Ever wondered how to supercharge your study sessions? Well, it's all about creating killer notes. Effective note-taking isn't just about jotting down facts, it's about organizing information in a way that's easy to understand and remember. Especially during CSIR NET exam preparation phase, notes serve as a handy resource and can be used for quick reference, self-testing, and identifying areas that need further review. Hello CSIR NET Life Science enthusiasts, I'm Dr. Tanushree, and in this video, you will learn about how to take effective notes for CSIR NET Life Science exam. Let's first understand what is the importance of making effective notes. The advantage of making notes for CSIR NET Life Science exam are as follows. First, organization. Creating notes allows you to organize the vast amount of information covered in the syllabus into manageable chunks. This organization helps in better understanding and retention of concepts. Second, active learning. Writing down information forces you to engage actively with the material rather than passively reading or listening. This active learning process enhances comprehension and memory recall. Third, effective learning. Use of diagrammatic representations while making notes, either in form of flowchart, bullets, helps you memorize topics much effectively. Fourth, personalization. Everyone's learning style is different. By making your own notes, you can personalize them to suit your learning preferences, whether it's using diagrams, charts, mnemonic devices, or summaries. Fifth, retention. Research shows that the act of summarizing and rewriting information helps in better retention of knowledge. Notes serve as a concise recap of what you have learned, making it easier to review and revise later. Sixth, clarification. While making notes, you may encounter concepts or topics that you don't fully understand. This prompts you to delve deeper into those areas, leading to a clearer understanding of the subject matter. Seventh, efficient revision. During the revision phase, having well-organized notes saves time and effort. Very much beneficial and easy for last-minute revisions. You can quickly review key points without having to go through entire textbooks or lecture material. Eighth, portable study material. Notes are portable and can be carried out easily. Ninth, building confidence. Creating comprehensive notes gives you a sense of accomplishment and confidence in your preparation. It also provides a roadmap of your progress, helping you identify areas of strength and weakness. And 10th, long-term learning. Making notes is not just beneficial for exam preparation, but also for long-term learning. The process of summarizing and synthesizing information helps in building a strong foundational understanding of the subject, which can be valuable beyond the exam. So, how you should begin with notes making? The very first point is understand the syllabus. Familiarize yourself with CSIR NET Life Science syllabus. Identify important topics. So while making notes, cover important topics first. Then gather study material. Collect all the relevant study materials such as reference books, lecture notes, and online resources. Use multiple sources. Don't rely on just one source. Consult 2 to 3 reference books or online resources to get a comprehensive understanding of each topic. Then create an outline. Divide the syllabus into manageable sections or topics and create an outline to organize your notes effectively. And make sure you take concise notes. While studying each topic, focus on understanding the key concepts. Take concise notes using bullet points, diagrams, flowcharts, mnemonics to make the information easier to remember. So how you can prepare notes for CSIR NET exam? You can prepare notes for CSIR NET exam in tabular form, in form of flowchart, diagrams, graphs, mind maps, mnemonics, and even you can create a formula sheet to revise and learn the formulas frequently. So let me give you certain examples. So here I have listed few topics from every unit of CSIR NET Life Science syllabus that you can study in tabular form. So from unit 1, you can study about charge and pK value of every amino acid in tabular form. Then you can learn about A, B, Z form of DNA. So you can make a comparative table to learn about these different forms of DNA. Then to study protein structure, primary structure, secondary, tertiary and quaternary structure. So again for that you can make a comparative table. Comparative table can be made for glycolysis and gluconeogenesis, to study glycolysis and gluconeogenesis. Then to study about enzyme inhibition like competitive, non-competitive and uncompetitive. And even you can study about the enzymes involved in protein digestion in tabular form. Then from unit 2, you can study about lac operon, mostly regulation in marrow diploid condition. Even you can study about cytoskeletal proteins. So you can make a comparative table of cytoskeletal proteins, inhibitors and motor proteins, and you can learn from the comparative table. Then from unit 3, you can study about the enzymes associated with DNA replication, so both in prokaryotes and eukaryotes. You can learn about factors associated with transcription. So you can mention the factors in the tabular form, and you can mention the functions of all the factors involved which are involved in transcription. Then even you can study about inhibitors of important enzymes along with their mechanism, and you can also list the pathogens and the diseases associated with them. From unit 4, you can make a table for B and T cell, and even you can make a table for cell adhesion molecules, oncogenes and tumor suppressor genes. From unit 5, you can study about different type of stem cells, speciation types, fate of germ layers, types of egg and cleavage pattern, and even genes involved in ABC flowering. You can learn it in a tabular form. And in unit 6, from unit 6, you can study about plant hormones. So I'll give you some samples. Some samples I have mentioned here, just to show you that how you can make a table for these topics, some of the topics which I have mentioned in the previous slide. So like to learn A, B and Z form of DNA, you can make such a comparative table where you can mention about the attributes, like what is the helix sense of A form, B form, Z form, then rotation per base pair, and what is about how many rise or per turn of the helix is there, or rise per base pair along axis is there, what kind of puckering is present, sugar puckering is present. So this is how you can make a comparative table. Similarly, to learn about the DNA replication in prokaryotes and eukaryotes, so you can make a comparative table. So you can mention about how many polymerases and what polymerases are involved in replication, DNA replication of prokaryotes and eukaryotes, their function of polymerases you can mention, and even you can mention about the origin of replications in prokaryotes and eukaryotes, and so on. So comparative table is the best approach and the best way of learning and revising these topics. So make sure that whenever you are making notes for these topics, you make it in a tabular form. Also, like to learn about protein factors required for initiation of translation in bacteria and eukaryotic cell, you can make a table. So here you can see that you can mention about the factors and their function. So in one column you can mention the factors, and in another column you can mention the function of the factors which are required, protein factors required for initiation of translation in bacteria and in eukaryotic cells. Then similarly, you can make a table for proteins required for initiation of transcription at RNA polymerase 2 promoters of eukaryotes. So here you can see that transcription proteins are mentioned in one column, the number of subunits in another, and even the functions are mentioned. So for initiation, these are the transcription proteins required. For elongation, these are the transcription proteins required, and their respective functions are also mentioned. So you can see that these are the key points that you have to keep in mind, like even for solving the questions based on these topics, these are the information you need to recall. So table help you in learning and recalling, in revising, and also at the time of exam you can easily recall if you learn these about these proteins in this way, like in a form of table. You make a table and you learn. Then from unit 8 you can study about mutagens or nature of mutations induced by them in a form of a table. So you can mention the mutagens, and you can mention what mutations, what is the nature of mutation is induced by them. Then from unit 9 you can study about features of different phyla in animal kingdom. Like here you can see that features of different phyla in animal kingdom is mentioned. So about Pisces, amphibia, reptilia, aves, mammals. So what kind of respiratory organs they have, heart is, how many chambers in the heart, then what kind of skin they have, whether they have skin covered with scales, or they have mucus glands in skin, or they have skin covered with feathers. So all the features are mentioned here. So it's very easy to compare and learn, and when you compare, you learn faster. So make a comparative table. Then from unit 10 you can make a table for species interaction for different ecosystems, and even you can make a table for threatened plant and animal species. So you can mention the names, the examples, and whether they are, like, you can mention their status, that they are extinct or extinct in the wild or vulnerable or near threatened. Then from unit 12 we can study about function of weird genes as shown over here. So the different weird genes are shown and their function are mentioned. So again, it's very easy to remember, to learn, to revise about weird genes in a table form. Then from unit 13, comparative tables are very much needed. Like you can compare different immunological techniques like RIA, ELISA, because most of the time, like I get a lot of questions from the students, and they mention that, ma'am, we often get confused while learning the techniques. So best approach to learn the technique is make a comparative table. So you can compare like RIA with ELISA, or circular dichroism spectroscopy with NMR spectroscopy. You can compare light electronscope with electron microscope, and so on. So I will suggest that especially for unit 13, comparative table is the best approach to learn about the techniques. Then even it's not that you have to only make the tables. For some topics even you can make the flowchart. So specifically like biochemical pathways, especially from unit 1, can be remembered in the form of flowcharts. Then even you can make a flowchart of proteins and factors involved in cell cycle regulation from unit 2. You can make a flowchart of transcription factors and steps involved in transcription and translation from unit 3. Then from unit 4 you can make a flowchart for signaling pathways. Similarly from unit 6 also you can make a flowchart for the pathways like C2, C3, C4 cycle, dark reaction, light reaction. Even you can make a flowchart of plant secondary metabolites. And from unit 9 also I will suggest that to learn about plant and animal classification, flowchart is the best approach. Like it's best way of learning these topics is that you make a flowchart. You make the notes in the form of flowchart. So again, for the flowchart, I'm showing you certain samples. So like here you can see this is a flowchart of plant secondary metabolite. So it's very easy to learn about plant secondary metabolites, like they are terpenes, phenolics, N containing compounds, S containing compounds, right? And even the examples are mentioned. Then similarly, if you're learning about plant classification, so it is also again very easy to remember, like starting with when the plants, they do not have differentiated plant body, so we call them as thallophyta. Then they have differentiated plant body, but they don't have, some have no vascular tissue, so they are bryophyta, and some have vascular tissue. So again, the plants which have vascular tissue but do not produce seeds are pteridophyta, which can produce seeds are phanerogams, and so on. Then even you can see, you can learn about biosynthesis of amino acids, so you can make a flowchart for it. And even to study about operation of rod photoreceptors, you can make a flowchart like this. So don't you think it's very easy and easy to remember and easy to learn when you make the notes in this form, in a form of a flowchart? Even you can make notes in forms of diagrams, especially for the topics like cycles that you study in unit 1, like urea cycle, or in unit 6 like C2, C3, C4 cycle, and more. So you can draw the cycle, and below it you can mention the important points associated with the cycle. Then even to study about genome organization, learning through diagrams is the best approach. So you can make a diagram of nucleosomes, histones, and even types of order of packaging you can better understand with diagrams. Then from unit 5 to study about ABC flowering, ABC model, again you can make a diagram and learn. Then to study about cladistics, to study cladograms, phylogenetic tree, to study about monophyletic group, polyphyletic group, paraphyletic group, to understand synapomorphies and plesiomorphy. Again, diagrams, making the diagrams and learning is the best approach. Then to understand about chromosomal alterations, to understand agrobacterium mediated gene transfer process from unit 12. Again, you can make a diagram for such topics and you can learn from them. Then to understand gene cloning, to understand about the vectors, about the restriction digestion, to understand about the process of techniques like ELISA, about radioimmunoassay, flow cytometry. The best approach is that you make the diagrams and understand the techniques, process or the concept behind it. And even to understand about instrumentation, that how UV visible spectroscopy works or how the light is transmitted through the spectrometer and reaches to the sample, to understand mass spectrometry, that how it works. The best approach is that you make a diagram. So here I have added few samples that will help you understand how you have to make the notes in forms of diagram. So like this is a diagram, a schematic of octopine type Ti plasmid. So you can see it's a well labeled diagram and it's quite self-explanatory. You can understand what all genes are present in this plasmid, right? You can understand about the left border, the right border. Then to understand about cladogram, to understand phylogenetic tree, to understand about different groups, paraphyletic, monophyletic, as I mentioned earlier, the best approach is that you make a diagram and study. Then for techniques also, like to understand the process of indirect ELISA, that how the antigens are coated and how we are when we are adding the antibodies and when we are adding the secondary antibodies. So for that also diagrammatic representation is the best approach. And even to understand how a particular techniques works, again for that it's best to make the notes in form of diagrams and study from it. Even you can make mind maps. Mind map is a diagrammatic representation where the central idea is broken down and each branch helps you understand an essential part of the concept. For example, to study photosynthesis, to study respiration, to study about the topics in unit 7 like female reproductive cycle, you can make the mind maps. So here you can see the mind map is about female reproductive cycle. The entire process is mentioned here. And then you can see that ovulation, menstruation, proliferative follicular phase. So even subpoints are mentioned for these main topics. Even to study about phytohormones, the examples of phytohormones, the name of receptors, the nature of receptors and signaling pathways, you can learn in form of the mind maps. So all you need to make a mind map of the phytohormones and you can mention the topics and subtopics or the information about the topics and easily learn from mind maps. Then even you can make graphs. So you study about proteins and factors involved in cell cycle regulation in unit 2. So you can note down the proteins and factors involved in cell cycle regulation from text and you can design it in a graphical way. Then prey-predator relationship. So in unit 10, you study about the Lotka-Volterra model, also referred to as predator-prey model, and it is a well-known mathematical tool for studying the dynamics of interacting biological populations, with a particular emphasis on the interaction between predator and prey species. Then even to learn about island biogeography. So island biogeography explains that an island's biodiversity and the number of species it supports are influenced by both its land size and extent of its isolation. So even you can remember, you can learn about these topics by making notes in a graphical form. Then to learn certain topics from unit 10 and unit 11 specifically, you can take help of mnemonics. For example, if you want to learn about geological periods, you can make a mnemonic. Camels often sit down carefully, perhaps their joints creak. So here C of camel is for Cambrian, O is for Ordovician, S for Silurian, D for Devonian, C for Carboniferous, P for Permian, T for Triassic, J for Jurassic, and C for Cretaceous. Don't you think it's so easy? When you make a mnemonic, it's very easy for you to learn about the geological periods. Do let me know in the comment section what you think about it. Then even to learn the terms, to learn several terms from Unit 5, Unit 8, Unit 11 and 12, you can note down the terminologies. Like if you want to learn the evolutionary terms, about the bioremediation, about the genetics terms like about alleles, about backcross, about test cross, codominance, incomplete dominance, and even to learn about the terms from Unit 12 like bioremediation and so on, you can note down the terms. And then even you can make flashcards for topics like carbohydrates and proteins from Unit 1 or photosynthesis or respiration from Unit 6. See, kindly note, you can make notes in any form, whichever form you find it useful or whichever you find it most appropriate according to your learning style, you can use that method. So here I am just giving you the examples that this approach you can use while making the notes. So even you can make flashcards, and also there are several topics given in the syllabus from which the numerical questions are asked. So certain formulas are there which you have to learn. So for that you can make a formula sheet. You can take a paper and you can mention all the formulas unit wise on that paper, and you can stick it on the walls of the room so that whenever you enter the room, you can see them and you can revise them and learn them as soon as you see them. So here are some of the formulas that you can for which you can make a formula sheet. So from Unit 1 you have several formulas like Hasselbalch equation is there, Michaelis-Menten equation is there, then how you define pH, how you define pOH. Then from Unit 2 you study about microbial growth kinetics. So even there is a formula to calculate colony forming unit, CFU. Then from Unit 4 here are some of the formulas like what are the in order to determine the possible number of combinations of VDJ and VJ joining, what formula you are going to use. Then in Unit 6 plant physiology, you have a formula for calculation of water potential, for calculation of ATP from reducing sources, NADPH2, FADH2. Then respiratory equation formula is there, and even the formula for quantum yield of photosynthesis is there. Then in Unit 7 animal physiology, you have a formula for cardiac output, net filtration pressure. Then in Unit 10 you have a formula for mark recapture method. And even from Unit 8, I missed Unit 8. So from Unit 8 you have several formulas given like how to calculate recombination frequencies, and even recombination frequency formula for ordered and unordered tetrad. Then Lodge score is there from Unit 8, and many more formulas are there. Then from Unit 11, so again from Unit 11 there are certain formulas which you can remember, like Hardy-Weinberg principle, that is the most important you can remember. Then from Unit 13 you have a formula like you have to learn about Beer-Lambert's law, you have to study about how to calculate the half-life. Then you have the formula for calculating the probability of finding a restriction site, and so on. And lastly, I would like to give you certain preparation tips as well. So as I mentioned earlier, that make sure that notes you make should be concise. Notes making doesn't mean that you simply note down everything line by line from the reference books or whatever resources you are referring. Notes should be point to point, it should be concise, and make sure that you mention the important points only in the notes. You can while making the notes, you can make use of bullets or numbering, and try to make the notes colorful because it helps you. The notes will be then you find them attractive and easy to understand if you use colors and even diagrams to visualize complex ideas. And don't forget to include examples for better comprehension. Like for example, if you are studying about radioactive decay, so this is a notes from our concept notes of Biotechnica. So like here you can see that like if you are studying about radioactive decay, so let's say alpha decay you are studying, right? So important points I have highlighted in red, like alpha decay involves loss of 2 protons and 2 neutrons, and some more important points I have mentioned. And even I have mentioned the example so that you can actually relate that how 2 protons are lost and 2 neutrons are lost, how atomic number decreases by 2 or mass number decreases by 4. And even you can use different colors, like you can highlight the important points in different colors, and the less important points in different color you can mention. So with this, I have come to an end of this video. So the takeaway message is that in your quest for success in CSIR NET Life Science exam, meticulous note-taking is your ally, helping you navigate the academic terrain with confidence and clarity. With well-crafted notes, you will not only breeze through your studies, but you will also ace the CSIR NET Life Science exam. So, what are you waiting for? Grab your pen and paper or keyboard and screen, and let's start crafting notes to ace the CSIR NET Life Science exam. Thank you.